Hello, my name is Nicholas Handfield-Jones. I'm a first-year PhD student in neuroscience in the lab of Dr. Penny McDonald and co-supervised by Dr. Ali Khan and Dr. Adrian Owen. Thank you for expressing interest in my poster for OHBM 2020. By watching this short video, my hope is to walk you through the work that we have done on dopamine connectomics to answer a simple but overlooked question. Are dopamine circuits as simple as we think they are? The figures that you see on my poster are called connectograms. At first, these diagrams can be quite confusing. To understand them, it is helpful to understand the background behind the current conventional dopamine pathway heuristic and why we think that it is oversimplistic. To start, let us review the anatomy of the dopamine system. In its simplest form, the dopamine system comprises two dopamine-producing nuclei and a host of target brain regions that receive dopamine via dopamine receptors. The dopamine-producing nuclei are located in the midbrain. The larger of the two, the substantia nigra pars compacta, shown here in blue, can be abbreviated as the SNC, while the smaller of the two, the ventral tegmental area, shown here in red, is called the VTA. According to conventional paradigms, dopamine from the SNC projects to the dorsal striatum, or DS, which is made of the dorsal bulk of the caudate putamen. This pathway is called the nigrostriatal pathway, and is thought to mediate movement and decision-making. On the other hand, the VTA is thought to project dopamine to the ventral striatum, or VS, which is formed of the ventral aspects of the caudate putamen and the nucleus accumbens. This pathway is termed the mesolimbic pathway and is thought to be involved in reward processing. Here, we call this paradigm the conventional pathway heuristic. Importantly, the conventional wisdom behind this heuristic is that these pathways are entirely distinct. In other words, the SNC does not overlap to project to the VS, and the VTA does not overlap to project to the DS. However, evidence has been accumulating, even as early as the 80s, to suggest that these circuits are more complex than their conventional pathway heuristic. In this 1994 study by Lind-Balta and Haber, for instance, retrograde tracers were placed in the VS, while the majority of tracer was found to originate in neurons in the VTA, as expected, an amount was found in the SNC. On the other hand, this 1983 study by Parent and colleagues placed retrograde tracers into the DS, and it was found that while most originated in the SNC, some were found in the VTA. These two studies were performed in non-human primates, but these findings have been found in rodents as well. In humans, the extent to which the conventional pathway heuristic exists is unknown. One method to potentially gather information about these pathways is through diffusion magnetic resonance imaging, called DMRI. DMRI is a technique that measures the strength and orientation of water diffusion within each voxel of the brain. A voxel can be thought of as a 3D pixel. The diffusion of water is measured because it represents the larger neural tracts, as water will flow along axons. A DMRI scan of the brain allows us to perform a computational technique called probabilistic tractography. In tractography, we can select one brain region to be a seed region, which acts as the origin point, and other brain regions as target regions, which act as endpoints. Then we can compute 5,000 probabilistic streamlines which follow the calculated water densities along the path of least resistance. These are calculated from the seed region to target region to estimate the connectivity density from seed to target region, based on the number of streamlines that pass through each target. In our experiment, we used DMRI data from the Human Connectome project to investigate these questions. We set seeds to be in the SNC and VTA to measure the percentage of connections to and from these regions to the DS and VS. We also set seed regions in the DS and VS to measure the percentage of connections to and from these regions to the SNC and VTA. Doing these measurements in both directions gives a more complete picture of how the SNC and VTA connect with the DS and VS. Following these measurements, we performed repeated measures ANOVAs to compare the connectivity percentages between the SNC and the VTA within each type of seed to target test. First, what happened when seeds were placed in the SNC and in the VTA? In the following set of slides, we will show you the results we found. In the accompanying figures, the width of the line represents the connectivity percentage. First, when the target was in the DS, we found no significant difference between SNC and VTA. In this image, we can see that the thickness of the lines are the same. On the other hand, when the seed was placed in the DS and the targets were the SNC and VTA, we found that the SNC target was significantly greater than the VTA target. With respect to the VS, when it was the target, we found that the connectivity of the VTA to and from the VS was greater than that of the SNC. However, when we placed the seeds in the VS, we found that the connectivity of the VS to and from the SNC was significantly greater than the connectivity to and from the VTA. In sum, the results we found are at odds with the conventional pathway heuristic. If that heuristic truly existed, we would have expected to find that the connectivity values were distinct. However, we found an overlapping pattern. This can be seen when seeds were placed in the SNC and the VTA, and when the seeds were placed in the DS and VS. We summarize these findings in the connectograms that you see on the poster. In these figures, the outer circles represent the brain. The outer circle is subdivided into sections representing different brain regions. The lines in the middle of the circle represent the connectivity between each brain region, where thicker lines represent thicker connectivity density. Importantly, the image on the left shows when the seed was placed in the SNC, and the image on the right shows when the seeds were placed in the VTA. We can see that in both cases, there was the same amount of connectivity to the DS, which is contrary to the conventional pathway heuristic. Thank you for listening to this summary of my poster. Our hope is to continue examining these pathways using the Human Connectome Project. We have found that dopamine pathways are not as simple as conventionally thought in living humans. This has implications for further investigations into dopamine and dopamine-mediated disorders. I would like to thank my supervisors and my colleagues for help with this project. If you would like to get in contact with me to ask any questions, please send me an email at nicholas.handfield@gmail.com. Thank you.